Hello everyone and welcome back to Medinotes from Medicos. The topic that we're going to deal today is PCR from microbiology. The objectives we're going to deal over here is introduction, the principles, steps, types and applications. So what is PCR? PCR basically stands for polymerase chain reaction. You might have studied in biochemistry as well as during your 12th standard. So it is a method that is widely used to create billions of samples of copies of a specific type of DNA. This method enables a detailed study using a very small fragment of DNA. So what is the principle behind this? Actually, this is based on an enzymatic replication of the DNA. Here what happens is a short fragment of the DNA of interest serves as the template, which via the chain reaction um, is amplified using the primer mediated enzymes. Now what happens is the DNA polymerase synthesis a new strand of DNA complementary to the DNA template DNA, and then the DNA polymerase they add new nucleotide, but only at the pre-existing 3-OH group only. Now, what are the steps? So basically, there are three steps involved in polymerase chain reaction. One is the denaturation, second is the annealing, and third is the extension or the elongation. So you can see the picture over here clearly. This is a small fragment of the DNA. This is the original DNA that is to be replicated. This is a nucleotide. This is the DNA primer. So at the temperature of 94 to 96 degrees Celsius, separation of the DNA occurs to form templates. Okay, now the templates is formed over here. Now at the temperature of minus 68 degrees Celsius, there occurs annealing. Annealing means it goes and attaches to the other DNAs. Now at the degree of 72 degrees Celsius for 2 minutes, it undergoes extension or the elongation. This process goes on, and so we get billions and billions of copies. So what are the types of PCR? Now there were initially 4 types of PCR. This too has been added recently. So what are it? RTPCR, Quantitative PCR, Nested PCR, Multiplex PCR, Clip-based TrueNAT, Cartridge-based Gene Expert or CBAT. Now we will learn a brief about all of this PCR. So what is RT-PCR? RT-PCR stands for reverse transcriptase PCR. So what is it? It actually here the RNA is used as template to obtain the complementary DNA strand with the help of reverse transcriptase. That is the RT enzyme. This is a modification method of RT-PCR and it can only amplify double-stranded DNA sequences. Now we'll be going to qPCR. Actually, qPCR is also known as the real-time PCR. Here before it was named initially as RTPCR, but because it was having a problem or confusion between reverse transcriptase and real-time PCR, they shifted it as quantitative PCR or qPCR. So what is this? By the name of quantitative itself, we understand that it is based something on quantifying. So it quantifies the PCR by monitoring the amplification process while the PCR is ongoing. And here what happens is it estimates the pathogen in the DNA load of the test specimen, and then the samples can be processed for rapid detection of specific DNA in cases like hepatitis B DNA viral load. And then we have the detection. How is the detection done? The detection can be done in two ways. One is by using a non-specific fluorescent dyes like SBYR green. How does this work? It intercalate with the double-stranded DNA and then get synthesized. We can also use fluorescent reporter signal strength. So in fluorescent signal strength, reporter signal strength, this is actually directly proportional to the number of amplified DNA molecules. The next is nested PCR. So this is used to prevent non-specificity. Sometimes what happens is this DNAs would interchange with each other and the specific DNA would get manipulated by others. So this one we would be using as a second set of specific primers in successive cycles to amplify a secondary target within the first target. So in case such things occurs, it goes and acts with that one and it forms a different one. so that we get the specific DNA that we want. Next would be the multiplex PCR. Here the primers that is used is more specific to more than one pathogens and can be used simultaneously. This is clip-based TrueNAT. So this was the method that was recently used for detection of SARS-CoV-2 during the COVID pandemic of 2019. The next is CBNAT. CBNAT is also known as Gene Expert. It is used widely for the detection of mycobacteria. Next is, what are the applications? So this is used for the diagnosis of infectious disease, for example, hepatitis B, genetics of neoplastic diseases, forensic investigations, archaeobiological studies of ancient specimens, examination of phylogenetic relationship in evolution. So that was today's topic. I hope you like this video. If you like this video, don't forget to like, share and subscribe to the channel. We'll be coming with more videos. Till then, stay tuned and happy learning. Bye.